Now let us move to the third part of biomolecules. Here we are discussing about fructose. Fructose is a ketohexose and it has the minus optical activity, that is levorotatory, and that is why it is represented as D minus fructose. Its molecular formula is isomeric with glucose. Both of them are, that is glucose and fructose are isomers, but glucose is aldohexose. Take down, it is aldohexose, whereas fructose is a ketohexose. In fructose, the potential carbonyl group is ketone, whereas in glucose it is aldehyde. Otherwise, the structures are almost similar. Here, the fructose has the furanose structure. Furanose means 1, 2, 3, 4, that is a 4-membered ring here. 1, 2, 3, 4, 5-membered ring. And the pyranose is a 6-membered one, inclusive of that heteroatom, that is oxygen. As I already mentioned, the ring formation is between 2nd carbon atom and 5th carbon atom. So that's why you are able to get the furanose. And it's called fructofuranose. So this is alpha fructo D minus fructofuranose. Because of the rotation of OH, here we can have, that is alpha furanose and beta fructofuranose. So likewise we have two types of isomers. Take down. The same structure, but CH2OH, C double bond O, and this involves in the hemiacetal ring formation with the 5th carbon atom. This is 1st carbon atom, 2nd carbon atom, 3rd, 4, 5. So between 2nd and 5th carbon atom, the hemiacetal ring is formed, and in fact the CH2OH is shown here. Okay, so when the OH is on the right side, then it's called alpha. When the OH on the left side, it's called beta. Please draw the structure. Similarly, we can draw the Haworth structure for sucrose. So it is based on furan. So furan like carbohydrate, this is known as fructofuranose, alpha D fructofuranose. As I already mentioned, the OH is downward, that is alpha. When it is reversed, that is upward, then it is beta. So alpha D minus fructofuranose, beta D minus fructofuranose, and this is the Haworth synthesis. So Haworth's structure. Now let us move to hydron.
4 minutes is it?
Right. So disaccharide. Disaccharides are the two monosaccharide units joined through glycosidic linkage. So let us consider sucrose. So sucrose is a combination of glucose and fructose. So when it is hydrolyzed, we are able to get D plus glucose with the optical rotation 52.5 plus 52.5. Whereas for fructose it is a minus 92.4. So it is a LEVO. So this is D plus glucose, this is D minus fructose. So the resultant is a negative one. But sucrose is a positive dextrorotatory. After the dehydrolysis, it becomes, the net rotation is LEVO, because this is plus 52, this is minus 92. That is because of the dominance of fructose also. So here the resultant mixture gives a LEVO rotation, and that is why it is called inversion of sucrose. Note this point, very important point. So why it is called inversion of sucrose? Sucrose as such is dextrorotatory, but when it is hydrolyzed, it is it is dissociated into D plus glucose with the 52.5 rotation and fructose, that is D minus fructofuranose with minus 92.4. So the resultant is negative rotations, that is LEVO rotation. So since there is a conversion of a dextro to LEVO, this reaction is known as inversion of sucrose. That indicates the rotation inversion, rotation angle inversion. So this is possible in the presence of a biocatalyst, and since it hydrolyzes the sucrose and convert it into an inverted rotation with the dominance of LEVO, and that is why this enzyme is called invertase. As I already suggested, the enzyme which is responsible for breaking the glycosidic linkage can be called ACE, because it is specific about OS, and that too is responsible for the inversion of OS. So it's called invertase. Then structure of sucrose. It is a non-reducing sugar. It is dextrorotatory. So as I already mentioned, the oxy linkage between the two units of disaccharide is called glycosidic linkage. So this will be broken during the hydrolysis, resulting the monomers. So draw the structure. Invertase, that is the biocatalyst. It's an enzyme that is responsible for inverting the optical rotation of sucrose from dextro to LEVO because of the splitting of the glycosidic linkage. So that is why it is called invertase, and that reaction is called inversion of sucrose. So sucrose here, it has got dextro rotation, that is positive rotation as the net one, whereas here, because of the difference in value and the higher value of levo, the net rotation of the mixer is levo, and that is why, since it is a rotating plus to minus, it is called levo, inversion of sucrose. And the responsible biocatalyst, that is enzyme that causes the inversion, is called invertase.
Okay.
Similarly, maltose. So the maltose is the combination of alpha D glucose and alpha D glucose. So by combining two alpha D glucose, two monomers of alpha D glucose, we are able to get maltose. It is connected through the alpha glycosidic linkage. In fact, I just reversed this. You know the structure of maltose, the glucose, where you have HOH, OHH, HOH, CH2OH, H. Now you take another unit of glucose and just reverse it, interchange, that is, you turn it 180 degree. So here you have to consider this one, H. So the removal of water takes place between, actually that is, there will be a OH group here, and here another OH. Now water is removed by the combination of these two, forming glycosidic linkage. So this O has gone here. Do you understand now? So this is known as glycosidic linkage. Next, cellobiose. In the case of cellobiose, it is called 2 units of beta glucopyranose. So beta, how do you do? That is, you take OH here, and as usual you can put here. Now HOH, and you can find the O is almost crossing like this. And that is why this cellobiose has the tendency for twisting, is it not? Here it goes as a linear polymer, or linear, in that case a dimer only. Now several cellobiose can result in cellulose. Do you understand? And that is why it forms a fiber-like structure. And as you know, these cellulose fibers are very popular. Okay. So what is the maltose? It is the dimer of alpha D glucose through the alpha glycosidic linkage. And what about cellulose? Cellulose is the disaccharide of 2 units of beta glucose, that is beta glucopyranose. And here how the link is developed through the beta pyranose only. Similarly, we have the milk sugar, which is nothing but lactose. In fact, some of the young kids, they are not, that is, they are allergic to lactose, and there will be some dysentery like disease when you take milk in childhood. So that disease is known as lactosis. So generally doctors advise not to take milk. It is because that immediately results in lactosis. Okay, so they advise equivalent cereals. Okay, இந்த திணை மாவுன்னு சொல்லுவாங்கல்ல, அந்த மாதிரி கொடுப்பாங்க இன்ஸ்டெட் ஆஃப் மில்க். சத்து மாவுன்னு சொல்லுவாங்க, சத்து மாவு அப்படிலாம் சொல்கிறாங்கல்ல, அது மாதிரி கொடுப்பாங்க. தென் மில்க் சுகர். ஸோ திஸ் இஸ் lactose. So the lactose is the combination of beta D galactose and beta D glucose. Look at the structure of galactose. So it's a combination of beta D galactose and beta D glucose. That is lactose. So these are all something concerned with the disaccharides. And similarly we have polysaccharides. They are the starch and cellulose. So large number of monosaccharides results in macromolecules. So, and in fact such type of polysaccharides they act as a food storage for structural materials. Starch is the polymer of alpha glucose, and it has got two parts, amylose and amylopectin. Now let us explain the difference between amylose and amylopectin. Amylose is a simple unbranched structure of the glucose units, that is several glucose units, whereas amylopectin is a branched structure, strained structure. So that makes the difference. Since it is unbranched, it is soluble in water, whereas amylopectin is not soluble in water. It will absorb water and swells up. Then it has alpha 1,4 glycosidic linkage. Alpha 1,4 glycosidic linkage means what? Now this is the alpha glucose. Are you able to understand? This is the alpha glucose. Here it has got the link between 1 and the adjacent fourth carbon, okay, of the adjacent glucose. So that results what? Alpha glycosidic linkage. So likewise it goes on, a linear structure. It gives a blue color when it is treated with a dilute iodine solution, whereas amylopectin it forms an yellow or orange with iodine. The molecular weight ranges from 10,000 to 50,000, but here it is much more than that. That is above 50,000 up to 1 million. Now let us look at the structure of amylopectin. This is the structure of amylopectin. As I already mentioned, see, that is, you have got alpha glucose, and we already know it is connected through the alpha glycosidic linkage. All these things we know, but it is since it goes on as a linear linkage, now there is a cross linking like this, and that's why it's branched. Are you able to understand? And this link can take place between 1 and 6 glycosidic bond. So this is the 6th carbon atom, this is the 1st carbon atom, so that is why it's called a 1,6 glycosidic linkage, and this is the regular 1,4 glycosidic linkage. So there are two types of glycosidic linkage, and which type of bond is responsible for branching? That is 1,6. So this is responsible for branching, is it not? And this one for linear. Okay. So in amylopectin there are two types of glycosidic linkage. One is 1,6 alpha 1,6 that results in branching, and what about the other one, alpha 1,4, that is also available in amylose. Okay, so that is responsible for the linear linkage, that is alpha 1,4 glycosidic linkage. The cellulose is something different. Here it develops beta linkage. Cellulose, a straight chain polymer, but it will be having a twisted nature, as we have seen in the cellobiose. So in cellobiose, as I already mentioned here, see that it is a beta linkage, is it not? And now likewise it goes on twisting like this, and that is the structure you are able to see. So instead of putting a linear one, I have drawn like this. So here you can find, you know very well, this is H, this is OH, means beta, is it not? So beta linkage. And what about this? This is H, this is OH, that is beta. So for every beta there will be a connection. So it goes on like this. So what type of linkage is present in between the monomers of cellulose? It is C1C4 beta glycosidic linkage. Do you understand this? Or it can be called a beta 1,4 glycosidic linkage. And you should know something about the glycogen also. What is glycogen? Glycogen is the carbohydrate that is present in animal body, that is stored in animal body. So it's a carbohydrate stored in animal tissues, animal body. So such animal starch is almost similar to amylopectin. It is present in muscles and brain, and also in liver. So they are called glycogen. Now let us summarize the importance of carbohydrates. See, let us start from starch. The starch consists of two types of structure, amylose and amylopectin. As I already mentioned, amylose is a straight chain polymer. Amylopectin is a branched one, cross-linked one. This is soluble in water, this is not soluble in water. So on hydrolysis, we are able to break the alpha glycosidic linkage. So it results maltose and maltotriose. Similarly here, and we use here amylase. Lase means the enzyme that is responsible for breaking the alpha amylose, and similarly we break with the help of the enzyme amylase, alpha amylase. So here the amylopectin gives maltose, maltotriose, apart from that alpha dextrin also. And these saccharides in turn on hydrolysis in presence of sucrase, maltase, glycoamylase, we are able to get glucose. Whereas here in the case of maltose, maltotriose, alpha dextrin, by using sucrase, maltase, then glucoamylase or isomaltase, these are all the isomers, we are able to get glucose. So there are various routes to prepare the glucose. And similarly from cane sugar, so cane sugar is nothing but sucrose, is it not? So as we have already discussed, the inversion of sucrose, that is possible by sucrase or invertase. So here we are able to get fructose and glucose. Then by lactase we are able to get galactose and glucose from milk sugar, that is lactose. Okay, so this is the overall summary about the different types of saccharides. Any doubt? So far we have discussed in detail about the carbohydrates. In our next session, I will be discussing the remaining part, that is amino acids, proteins, then vitamins, minerals, which are all the essential elements. So do you have any doubts so far? If there is any doubt, you can ask me.
Can you give reasons?
Okay, you prepare well and I will conduct after around 9:15. You must be ready before attending. You should not take it very lightly. You should be serious. If you are very serious about the quiz, then I am also ready. But without preparing, just for fun, you should not attend the quiz. Right?